Hi, is my screen visible to all of you?
Yes, sir, it's visible.
Yes, thank you. Thank you. Okay, so we will start. Okay, good evening, everyone. Thanks a lot. So today agenda is to conduct revision session only for week one and week two for ATB. And what we will do, we will go through the main concepts of week one and two. Thereafter, we will just see the PAs and GAs of week one and two. And thereafter, we will just take the questions from PYQs specifically to these weeks. Okay. And week three and week four, we might do either tomorrow or day after tomorrow, but definitely that we'll do before Friday, before Saturday, so that we have got ample time to prepare and revise our concepts. Is that okay?
Yes, sir.
Yeah. Okay. Okay. Good. So with that, so we go to the week one notes. Okay, so these are the week one notes. I hope my screen is clearly visible to all of you.
Yes, sir, it's visible.
Okay, so in the week one, what Professor he gave, motivation that why we should do this advanced logarithm algorithms for biotechnology. So it started with that, and then basic science. Knowledge of the DNAs, RNA, and the protein. So just to cover the DNA, main part of the DNA is inside the nucleus and it is in the chromosome. So we have got 23 chromosomes and every this 23 chromosomes, they got one part from the male and one from the female, from the parents. But there is a DNA also in the mitochondria also, which is outside nucleus. But in this course, we'll be studying basically mainly about the DNA, which is in the nucleus. So the DNA, what it does, the DNA does the transcription. It from that the RNAs are formed, and then these RNAs they come out of the nucleus, and then they produce the amino acids. The amino acids are nothing but the proteins. So the first part is known as transcription, which happens inside the nucleus, and the second part is known as translation, which happens in the outside the nucleus. So that is one part. The second part is that we have got these nucleotides. So this thymines, adenines, cytosine, and guanine. These are the nucleotides. So we have got four types of nucleotides. So and we say them A, G, C, T, and only thymine can it can react with the adenine, so AT, and the cytosine can do with the guanine, so CG. So it is the ATCG. And these nucleotides, how there is a long sequence in the genome, and depending on these positions of this, the various amino acids are formed. So that is this concept will come in the PAs and the GAs also, and there are questions in the PYQ also. So I have covered the nucleotides, translation, transcription. So this is one example of the RNA, and just for that, that RNA has got a oxy group, that is a OH in the uracil. So and the DNA does not have that. So DNA, that's why it is also known as deoxide, that it is without OH. So this is the transcription, translation, protein. Proteins can be of various types. Okay? The structures can be different. Like this is a primary protein structure, secondary, tertiary. So these are the amino acids which are there. Then the genetic code, okay, used in translation. This question is there in the GA also, and in both the PYQs, this question is there. So how these codes they translate to various amino acids. So it is like GA and then G, it will translate to this. If it is GCA or GCG, GCC, it will translate to this. So we can see that there is a lot of redundancy also here. GCG, GCA, and GCC and GCU, they are corresponding to the same amino acid. Okay, then there is a question in the PA or I think GA that where does this remarkable cellular diversity come from? That all cells they have got the same DNAs, but different cells, they will have different. So that is due to the reason that the subset of the genes and their products, that is the proteins, they are expressed in different ways from one cell to another. And this difference leads to the molecular content or the state of that cell. So that is the difference. And then the genes which are there, the cell definition by the molecular content or state, that like this gene 1, gene 2, gene 3 which are there. So in different cells, they might be having different relations. That gene 1 is leading to gene 2 and gene 3 both, and gene 2 and gene 3, then they're leading to gene 4. And in different cell, there might be a different structure. So on this also, there are questions on the PAs, GAs, and the PYQs. So that we'll see that concept there also. And this is the structure of one chromosome. So how the DNA is packed. So we can see it is a long sequence, and then it is structured in tablet kind of things. So they are padded up, and that's how the DNA is there. Already I mentioned we have got 23 pair of chromosomes, and in each pair we have got one from mother and one from father. And like all of us, all the humans, like we have got this ACGT, and we have got this genome length which is there. It is almost 1 billion. So that is a sequence of around 1 billion. And in every, in all the humans, the difference is roughly 1 in 1,000 locations only. So rest, it remains same. Only 1,000 locations which are there, which are known as, alleles. So that gives the difference in all of us, how we are different from each other. So this it is also referred as a mutation or a SNP, single nucleotide polymorphism. There is only one single nucleotide is getting changed. So if we have got 1 billion locations and we have got 1 in 1,000, it means in 1 million different ways we can be different. So this we have seen G1, G2 and all that. And this also can be expressed as an adjacency matrix or adjacency list. So for example, here we can see G1 is connected to G3 and both G2. So for G1 matrix is G2, G3. Then G2 is leading to only G3. So it is only to G3. And G3 is not leading to anyone. So it is 0, 0, 0, 0. Okay, and again, we can also, like we saw that in cell 1, the genes can be, having different structure. In cell 2, they can be having a different structure. So these cells also, they can be expressed in these ways. For example, in cell 1, what are the weights? For example, how much gene 1 is releasing how much mRNA? So here, these are the weights, 1, 3, 5, 0. So in cell 1, gene 1 is the amount of RNA is 1, here 3, 5, and 0. So this is how this becomes how we have been dealing in our MLP projects. So it is a data matrix where we have got features and we have got the data points. Now we come to, so with this I have covered the content in brief of week 1. Anybody, any question or any doubt till now? Okay, so I assume there is no doubt, so we can go ahead. So here I'll cover this important PA which is there, and this question has also come in the PYQ also. So this is the question that suppose a group of scientists have discovered the existence of alien life on a new planet. They are surprised to find that the alien life also follows the same central dogma. The difference is that alien life has 8 distinct nucleotides that make up the DNA sequence, and a whopping 200 distinct amino acids that make up the protein sequence. Suppose the ozone layer is not present in the planet's atmosphere and the alien life is exposed to ultraviolet and other radiation that can cause DNA. What would be your best guess for each codon's length? Okay, so I will explain it here, this part. So what they have said, like we have got A, T, G, C only, we have got only 4 nucleotides. Okay. And they have said that the codon length means that how many length will be there. For example, if I take N, so this is the codon length. Now, depending on the changes in this can lead to different amino acids. Is that okay? So see, if I've got, for example, ATGC, then I can have how many combinations I can have. And for example, the codon length is, for example, 4. So here I can have 4, here I can have 4, here I can have 4, here I can have 4. And this is the length of, for example, if I take length 5, for example. So I can have 4 to power 5. These many combinations I can have. So these are the number of amino acids, distinct amino acids, which I can create. Is that okay? Is that okay to everyone, this concept?
Yes.
Okay.
Yes,
So,
sir.
okay. So in this question, what they have given, that there are 8 nucleotides and there is a N. So here I can put 8 and for example, this is N. So it can lead to 8 to power N different amino acids. Okay. And they have given that there are 200 amino acids. So your 8 to power N, which is there, it should be greater than or equal to 200. Is that okay?
Yes, sir.
Okay. So now using like we can take the log of N greater than log of 200. Therefore, N bit of log N. No, there is some log of N should be greater than or equal to log of 200 divided by 8. Or we can directly check 8 to power 2, 8 to power 3. So when we take 8 to power 3, it comes around 216. So your N comes 3. Is that okay?
Should be 3. Yeah.
Yeah. Yeah. Okay. So this was the concept of this PA. Now we go to the next one. So that was other PAs. They were easy. You can go through them. Okay, just from the conceptual point of view, they were there. So the second, we will see the GAs. In the GAs, okay, they have just asked that what is the sequence? Okay, how all this thing happens. So the DNA serves as the template for mRNA production. That is true. So from the DNA, it does the transcription. From there, the mRNA forms. Then the mRNA comes outside the nucleus, and then it does the translation. So from DNA serves as the template for mRNA production. So that is the first step. The second is mRNA moves from the nucleus to the cytoplasm. This is the second step. In that cytoplasm, now the RNA bonds will do a specific codon. So this will transfer RNA bonds to a specific codon, and thereafter amino acids are formed. So this is the sequence how this entire process takes place. So one process is taking place inside the nucleus, and the second is taking place in the cytoplasm. Second is from theoretical thing, what is the relationship among DNA, genes, and chromosomes? So a chromosome is composed of a single long DNA molecule, and each chromosome contains multiple genes. So this is true. And this third option, which is there, it is also true. That DNA is composed of chromosomes, and each DNA molecule is made up of many chromosomes. Okay, then this is one is from the theory that all cells in our body have identical DNA because all cells have come from the same embryo cell. Then why does one cell look different from the other? This is the reason, because the different cells in multicellular organism may express very different set of genes. This is the GRN system, in which they have given that G1 regulates gene 2 and G3. So this is how we make the diagram by its protein product P1 and P2. And whereas gene 2 and gene 3 both regulate another gene 4 via their protein products P2 and P3 respectively. So just they have asked us to create an adjacency matrix. So this is how we make it. Quite easy. Then similar question which was there in the PA, in here they have given 6 distinct nucleotides that make up DNA and a whopping 100 distinct amino acids. Somebody's mic is not muted. Kindly mute it, please. So from here we find the N value. So this will come. And the last question was that suppose that the alien genome consists of 1 billion DNA bases, of which 1 million are SNPs. So this is from the same that 1 billion DNA bases are there, and 1 million are there which are here. The mutation is taking place. And if each SNP mutation can take one of the four forms, that four alleles which are there, so how many distinct alien genomes are theoretically possible? So this will become, we have got 4 and there are 1 million, so 4 to power 1 million. So that is the answer. Okay, so with this, I have completed week 1, and further we will go to week 2. So before this, before we proceed, anyone, any doubt till now? Anybody can pipe up, please. Okay, so I take it is okay with all of you, Aditya, Naman, Hitesh, Tushar?
Yes.
Yes, sir.
Okay, okay. So we go to now week 2. Okay, week 2 has got some formulas and some concepts. So in the week 2, basic concept is that how our cells which are there, they react. So the professor covered that cell on its own doesn't do anything. You know, this is the cell, for example, and it has got a nucleus. This is the nucleus. So here the DNA is inside, and here is a mitochondria. Here this DNA forms the mRNA. This mRNA comes outside, and here it forms a protein. But how this happen? So the scientists, what they have found out, that there is some trigger mechanism that this cell which is there, it keeps on observing its environment, that what is happening outside the environment. So this is the environment. Okay, and once it gets some trigger, and this is, for example, nucleus. So once it gets some trigger, then only this entire process of producing the different type of amino acids, it starts. So here it has got receptors. So this is a receptor. And here something is happening. Okay. So from here to here, there will be various paths. So it will be going through various RNAs. Okay, it will be going like this, like this, like this, like this, like this. And it is reaching here. So the concept wise, it was like that how to find the best path. So best path is one path which has got the probability which is there. So every this amino acids which are there, we have got from the experiments, we know that how they react to each other, that what is the weight. So we can give weight to every amino acid. So there are different amino acids here, here, here. So if you see, and these are the lengths again. So this is length of 1. So if you consider this diagram, so this one will be the most probable path because it will give the maximum probability. Okay. So there can be a different from here, 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 and here. So one which is having the maximum probability, that will be the simple path from this amino acid to this particle. So this is the main concept of your week 2. And how to find that, we'll see that. Okay, so this I have covered. And from there, okay, we'll go to the colorful part. Okay, we have studied in our, I think, MLF. So why does the cell need to sense its environment? I have told you that once it gets the, so this is how it happens. So from here, the gene and the ligand from here, so it is getting, and these are the different amino acids in the starting, or the mRNA is a protein that adds, and it reaches here. So this is the input and this is the output, where it is, and this is the nuclear membrane. Okay, and these are the various paths. Okay, now you can see from any part to any part, these are the different. So it can take this path also from here. It can take this path also. So the maximum, that most simple path will be the path which this will take. So can we find all the possible signaling pathways hidden in a large scale biological network? We need to know what a large scale biological network is there in the computational. So we see so much and how to find. So this is one example. So what we know that we know the protein interactions, and we know from the experiments on the previous that. So how we can do that, that if we know that this protein and this protein has got historically, for example, 0.6, this has got 0.2, this has got 0.3, and this is all in 0.4, and this is reaching here. So this is input and this is output. And there is another path, 0.4, and this is 0.2. So I can give you a quick hint, because these all are in decimals. So always you will find the path with the shorter length will always be the path which you can use to answer. Okay, so just find the length, and the length which is lesser will be the path to answer in the PYQs, in the quiz, in the GS. This all is mathematics. We have seen graph problem. We'll come to the the color point. Okay, and the main important is the logistics regression. So logistic regression, all of us we know logistic regression formula, that probability of is equal to is given by 1 plus 1 e to power minus summation of beta I into xi, where beta I starts from 0 to n. So by using this logistic regression formula, we can find the probabilities. And in both the PYQs, they have given question on this. So please remember this formula and how to use the calculator of our exam to do it more efficiently. So logistic regression, we have seen, given you the formula. So this same formula is given there. And then there is a brute force, and then we came to dynamic programming table. And the better solution is the color coding. So color coding is the one that if, for example, if I have to find path, so here, if I know that k, the length of the path, for example, I know it is 4. So what I will do, I will only take all the paths which are of 4 or less. Okay, I will discard any path which is more than 4. And how do I solve it? I solve it by the colorful problem. That if there are 4 nodes, 1, 2, 3, 4, so this will be a path only if these colors are distinct. Okay, and the formula for this is that the path will be colorful. So X is the number of colors, K is the number of nodes, and please remember this formula for the quiz 1. So probability that path is colorful is given by the formula xpk divided by x to power k. And this is nothing but, if you think mathematically, your total combinations which are there, it is x to power k, that in how many colors you can form this. If you put all colors here, put all colors here, put all colors here. So this is x to power k. And distinct colors, how you can choose, is x to power pk. So this becomes xpk divided by xk. This is another important. So this formula will come handy for your quiz. So now with this, I have covered the theoretical part. Before we move to the GS, any question, any query from anybody?
No.
Okay, thank you.
No, sir.
Okay. So is my screen clearly visible to all of you?
Yes.
Yeah. Okay. So this is the GA question 1. So they have given here receptors. So there is a receptor RA, RB, and RC. And they are receiving information about a harmful pathogen and transmit the information to one of the genes G1, G2, and G3. So these are the genes G1, G2, G3, and these are the receptors. So assume the time required to transmit information along any edge requires 1 unit of time. So they have given the time is 1. Answer the following questions with regards to the same. Assume that the number shown near each edge is the probability of the information transmission along that edge. And assume that these transmissions event along all different edges are independent of each other. So question 1, they are asking that what is the most probable path along with which gene G1 will receive information about the pathogen, takes so many units of time to transmit the information. So see, we see the gene 1, and what we do, we find its receptors. So receptors are RA, RB, and RC. So gene 1 can get information from only these three. So see, from the gene 1, if you see the shortest path, it is gene G1 to Z1, Y6, and to RA1. So this is the shortest and this is the most simple path. If you take any other path, it will be longer. For example, if you take G1, Z1, Y1, Y2, RD, for example. So this is longer. So this will take path length 3 and it will come from RA. Is that okay?
Yes.
Okay. Here I have given the full, calculations, but it is not required. As I covered that if you have got the length, if you can find the length, so the minimum length which is there, it will be the solution. Then second is that which of these paths have the highest probability of transmitting information? So it is simple mathematics. You have to put the probabilities, multiply them, and you will get, and then you can get the answer. Then we come to question number 3 and 4. So this question is from colorful. So in a random coloring of the nodes of the above graph, what is the probability that each of the following paths between A and G is colorful? Okay, and they have given, for example, a length 4 path using 4 colors. So here, see, if we see a length 4 path, so there can be only 2 paths of length 4, A, B, C, G, and A, B, E, G. And they have given color 4s. So directly we can apply the formula X is equal to 4, K is equal to 4. So probability that each of the following will be the colorful is XPK divided by XK, 4 factorial by 4 to the power 4. So that's how we get the answer. Question 4, in this they have given a length 4 path using 3 colors. So can we have that colorful? If we have got, for example, 4 nodes, 1, 2, 3, 4, but only 3 colors are there. So can we have colorful? One of the color will repeat. So probability is 0. Is that okay?
Yes, sir.
Yeah. So that's why this is straight away. You can cover it here from the conceptually only that this will be 0. Okay, then consider the problem of finding the best scoring simple path of the same. Suppose the color coding algorithm uses X colors, then we used to set K is equal to X. Can we set X less than K? Can we have colors? For example, this is K, 1, 2, 3, 4. Can we have X less than K? No, because then it will not be colorful. The same concept which just now we discussed. Is that okay?
Yes.
Okay. What is the probability? Straight away formula XPK divided by X to power K. And the question number 7, you are asked to compute the total number of K length paths from a set of receptors S to the set of genes T in the signaling pathway system G. Choose the best upper bound for W, Vx from the given options, where X is the set of X vertices. So what they have given that S and T and X. So we have got this S, these are the receptors, and these are the different receptors which are there. This is to the S, and here is the information, so T, and you have come here, for example, to some point X. Okay, so what is the max limit for any this path here? Now, what will be the max limit? So it will be X minus 1. So already we have covered one path, so it will be the X minus 1 factorials because it can go to different. Okay, consider 2 points x1 and x2 with the beta t x value of 10 and minus 10 respectively. Let px1 and px2 be the probabilities written by logistic regression. Which of the following is correct? So just we have to find the probability using the formula. Okay, and remember that formula is 1 plus e to power minus beta tx. Okay, so if this is plus, so this becomes minus, and if so just to cover the certain basics of the logistic regression, probability is equal to 1 plus e to power minus beta x. So as this x which is there, this beta x term which is there, if it crosses above somewhere 6 to 7, then this value becomes 0. Okay. And if this beta x which is there, it becomes negative, for example, then it becomes, if it is negative or it becomes minus infinity, so this value becomes 0 and then it is 1, because logistic regression is a non-linear. It works like this. So your probability, it is here. Oh, sorry. It comes from here and passes from 0.5. And it goes like this. So here is the probability 1 and here is the probability 0. This is x. Okay. Okay, what is which of the following is true for logistic regression? Very easy. It's for classification, and it returns the probabilities. This again from the logistic regression. What are the beta values? Beta values are beta 1 and beta 2 are positive values. Calculate the estimated using the formula of logistic regression. Easy. Okay, so this was the question, all the GS from week 2. So with this, we have covered the theoretical part and the GS of week 1 and 2. So before we go to the PYQs, any question from anybody?
No.
Okay, good. So now we go to the, I have taken all the questions from PYQs for these weeks. Okay, so this is the first question. We will solve it. Okay, so what they are saying, scientists, they have found an alien world where light follows the same central dogma principle. Their DNA consists of 5 different types of nucleotides, and so is their RNA. And amino acids are generated using codon of length 4. So this is codon, and it is got length 4. Also, due to some chemical structure of the nucleotide, any nucleotide can occur exactly once only in the codon. So this is one constraint they have given. Like in our this, it can repeat. For example, we can have A, T, C, G, A, like that. But here they have said that in any codon, the nucleotides which are there are 5 types. So we take an example A, B, C, D, E, and codon length is 4, and any nucleotide it can come only once. So see, from 5 I have to select 4. So in how many ways I can select? It is 5p4. So
Yeah.
it is 5 ways I can select. Okay. So just to give you an example, I can have A, B, C, D. I can have A, C, D, E. Oh, sorry. A, B, C, D. Then I can have A, B, C, E. I can have A, B, D, E. Is that okay? And then I can have A, C, D, E. And I can have B, C, D, E. These are the only 5 possible. In this, I can select the combinations. And once I've selected these 5 in this, now out of these 4, I have selected in 5 ways. So I have got, for example, I have selected A, B, C, D. Now, in how many ways I can select? So first part I can select, I can choose from 4. Second, I can select from 3 because they have to be distinct. Then here 2 and 1. So this is 4, 3, 12 into 20. So total number of ways is equal to 24 into 5 is equal to 120 ways. Is that okay?
Can you explain it once again?
Yeah. Okay. So they have given this constraint that due to some chemical structure of the nucleotide, any nucleotide can occur exactly once in a codon. So earlier, what we were happening that, for example, if it is not happening only once, then we can have 4, 4, 4, 4.
Yeah.
Okay, but it is saying that if I have selected, for example, A, then if I selected A, then A cannot come here. So in how many ways I can select? I can select either B, C, or D. So I got a choice of 3. And once I've selected B, then I can select only CD, 2. And here, if I selected A, B, C, then I'm left with only D. So these are the number of ways I can make the combinations from A, B, C, D. 4 factorial. Okay. And I have got 5 nucleotides and I have to have a codon of length 4. So in how many ways I can have? I can have in this form only. That is 5p4. I have to select 4 out of 5. So this is 5p4. This is 5 ways. So total number of ways is 5 into 4 factorial.
So first we are selecting and then arranging, right?
Yes, yes, yes. That's true. Okay. Okay, then we go to next question. Consider a GRN system with 4 genes G1, G2, G3, G4. The adjacency matrix is given below. How many genes are regulating G4? So straight away, what you can see, you can make a diagram also. This question also can come. So G1 is regulating gene 2 and gene 3. Gene 2 is selecting gene 3 and G4. So gene 2 is regulating gene 3 and additionally gene 4. Gene 3 is regulating only gene 4. And gene 4 is not regulating anyone. So how many genes are regulating G4? G4, you just see the incoming this. So 1 and 2. Is that okay?
Yes.
Okay. Okay. Next question is the figure below shows the genetic code describing the translation of an RNA codon into one of the 20 different amino acids. Use the figure below to answer the given sub questions. So see, these are the amino acids in outer this periphery, and these are the translations of the RNA strings. So RNA strings, they make in the sequence of 3. So first we take, for example, ACU. So you come to A, then you see C, we come to C, and then from C I go to U. And this corresponds to what? It corresponds to T. So I get T. Then I go to the second. Second is GAA. So I come to G, I go to A, and I go to A. So this corresponds to what? E. Then I come to ACG. So I come to A, then I go to AC, and then I go to G. So G is here. And this also corresponds to T. Then I come to GGU. So G, I go to G, and from G, I go to U. So this corresponds to this. This is G. And then is the UUU. So this is U, I go to U, from U to U, and this corresponds to F. So this is how we decode this. Is that okay?
Yeah.
Okay, so you take 3 combinations of 3 and then solve them. Okay. Okay, then second question is that if the amino acid leucine L is generated, then what is the probability that the codon encountered starts with uracil, that is U? Assume that the different positions of the codons are set to a nucleotide independently and uniformly at random. So what we can do, we can copy this and I will just explain on a new page. So it's a simple probability question. What they are asking, that what is the probability of L leucine, and it is coming from what? It is coming from U. L U of probability of LU. So probability of LU will be total number of LU and total number of L. That is the probability. And now you see LU. So L is coming from leucine. It is coming via G. So one is UUG and another is UUA. So we have got 2. So these are the leucines which are originating from U. So this number is 2. And Other leucine, if you see on the periphery, other leucine is here. And in how many ways it is getting generated? 1, 2, 3, and 4. So this becomes 2 divided by 2 plus 4. So this is 1 by 3 is equal to 0.33. Is that okay? Is this part understood?
Yes.
Yeah.
Yes.
Okay. Okay, then we go to the next question. Next question is receptor R receives information about a pathogen and transmits the information to gene via the following edges. R to Y1, Y1 to Y2, Y2 to Y3, Y3 to G. So R to Y1, it is given probability 0.02, Y1 to Y2 0.03, Y2 to Y3 0.06, Y3 to G is 0.01. So what is the total probability that the information has passed through this? We just multiply the probabilities and we'll get the answer. So it's simple mathematics. Next question is on the colorful problem. In a random coloring of the node of the underlying GRN network using 6 colors, what is the probability that this path is colorful? Random, and now they are talking about the same graph, which is there on the top. So see here, the number of nodes are there. So we know the formula that P of colorful is equal to X P K divided by X to power K. So they have given X is equal to 6 and K, if you see, is the number of nodes 1, 2, 3, 4, 5. K is equal to 5. So you just solve it and you'll get the answer, which comes 0.09. Is this okay? Everyone?
Yes.
So remember this formula, okay? Go. This question is slightly tricky because we have to read the theory also, the question carefully. It is a logistic regression problem. So what they have given, a potential cancer therapy drug Y was found to bind to certain cancer proteins in an experimental setup. Its binding was found to be based on its length L, presence of beta sheets, which is called by structure and which can take value 1 if the beta sheet is present, or 0 if it is a alpha helix, and presence of hydrophobic side chains, which is written as HSC. It is 1 if it is having side chains are hydrophobic, or 0 otherwise. So this is main part which you have to read. Using protein data from TCGA, a public database, logistic regression was performed to understand the drug behavior. The results are summarized below in a table. So what they are given for the length, the coefficient is beta is 0.9, structure is 0.3, HSC is 2.5. Okay, and for the amino acids, and here they have given now that it has got a alpha helix structure and it has got hydrophobic side chains. So what does it mean? If it is a alpha helix structure, so then this structure part which is there, it will be 0. Okay, if beta sheet is present, then it is 1. If it is alpha helix, it is 0. Similarly, HSC, if it is hydrophobic, then it is 1. So they have given it has got hydrophobic side chains. So this value is 1. Length they have given here 20. So this is L. And these are the coefficients. So these are the beta values. This is the constant. Okay. So they have given, so we have to straight away now fit the formula. So we know the constant beta 1, beta 2, beta 3 from here, and X values we get from here. Put it in the formula for logistic regression and we get the answer. So answer comes around 0.01. So probability of 0.01 means unlikely to bind because it is less than 50. 0.5. Is that okay?
Yeah.
Okay, thank you. Okay, next question is in a signaling pathway detection using the color coding algorithm, increasing the number of colors increases the probability of detecting the best scoring path as well as the running time of each color. So this is from straight away from theory professor had covered. However, just to demonstrate that the probability increase, you can take an example. You take X is equal to 4 and K is equal to 4, for example. K is equal to 4, then the probability comes around 0.093. As soon as you increase the number of color to 5, for example, x becomes 5, then your probability becomes 0.192. So as you increase the number of colors, your probability increases. And this second part is also there, that running time also it increase of each coloring run also increases, because now you got more number of colors, so you can select more, so it will increase your running time also. This is from the other PYQ, and straight away it has come from our question from the PA, that there are two distinct nucleotides that make up the DNA sequences and a whopping 600 distinct amino acids. So just to cover this concept again, so what they have written, that there are two distinct nucleotides. So there are two only, A, B only. We don't know the length. Okay, but okay, if it is N, so everything can take 2, because they have not given that they have to be different. So this total combinations will become 2 to power N. These many amino acids can become. Therefore, 2 to power N should be greater than or equal to 600. And from there, okay, you can calculate. Is that okay, this problem?
Yeah.
Okay. Okay, next one is very easy. Same G1, G2, G3, 4. They have given these diagrams, and we have to select the diagrams. So we know how to build this from here. So this is easy. The second part, the second question, already I have explained how to solve it. So we make the combinations of 3, 3, 3, and then follow them, and then you can write it down. So in this case, it comes IBP, VLTG. In this, the last one which is there, UGA, it is stop. So if we see here, you go to G and you go to A, and it is a stop. A stop means no sign. So there is no sign. So this is IBPVLTG. This we have solved again a very easy question using the formula only. And this one is from the theoretical part, that if we solve the color coding problem algorithm, which has got n nodes in the input graph, is assigned one of the k colors, the total number of DP table entries which are there. So this, please remember, this will be 2 to power k, where k is the number of colors, into n, where n is the number of nodes. So please remember this. This is from the dynamic programming, that how much you have to store, how many table entries will be there in your dynamic programming table. This is a logistic regression again. So here we have to read slightly carefully, and you will get the answer. So there he has given two features, x1 and x2. They have given beta 1 and beta 2 are the relative weights for this. Calculated the estimated probability, and they are given beta 0, beta 1, beta 0 is your constant, beta 1, beta 2 are these values, x1 and x2 are these values. So we have to straight away fit in the formula, and you'll get the answer. Okay, so with this, gentlemen, I have finished all the questions from week 1 and week 2. Any question, any query anybody wants to pipe up? I will answer that.
No questions.
Yeah, so it looks slightly easy, na? Week 1 and week 2. In fact, week 3 and week 4 are also easy, both of them. Shall then?
Yeah, it seems like the weightage of like first week is like higher than like last 2 weeks.
What, Naman? Can you come again, please?
I'm saying like it seems the weightage of first 2 weeks is like way higher than the last 2 weeks.
I think it is comparable. I think
Okay.
50-50. Yeah. So they'll come in 50-50. So maybe tomorrow or day after tomorrow, we'll discuss week 3 and week 4. So we'll follow the same pattern. We'll go through the important concepts, then we'll see the GAs and the PAs, and then we'll solve the PYQs. So with that, I think we'll be ready for the quiz. Is that okay?
Yeah.
Yeah,
This
absolutely
time
fine.
in this revision session, ma'am told that there might be some tricky questions. So they said that the core concept, you have to clear the core concept, then you can easily apply them.
Yeah, that's why Aditya, I'm covering the concepts. Okay, so you have to
Yeah.
understand what is happening. Okay, where the translation is happening, where the transcription is happening, where the receptor is there. Okay, so please understand those concepts again. And if you get the concept, then we'll be able to hack it.
Yeah. Yeah.
Okay. Chalo
Thank
then, bye. Have a nice day. Bye bye.
Yeah. Bye.
Thank you so much, Lalit sir.
Bye bye.
Good night.
Bye.